Hi, this is Dominic Reisig giving you a 2018 corn insect update. Sorry I can't be with you here today. I'd like to first acknowledge two graduate students in our program, Arun Babu, who's almost has his PhD out of our program, and Shawnee Gundry, who's working on a master's degree in our program, in our department. They contributed greatly to this presentation. Arun through his work for stink bugs on corn, funded by the Corn Growers Association and the Ag Foundation, and Shawnee Gundry, who's now being funded from a USDA NIFA grant. Today we're going to be covering stink bugs and earworms. This presentation is mainly the work of Arun, the PhD student who's been working on the corn. He's the real expert in this subject, and we're really excited to share with you today some of our changes for sampling and management of the stink bug. We'll be talking about the major species attacking corn. We'll talk about the susceptibility to stink bug damage, where you should look for it, and then how our sampling and management recommendations are changing as a result. So just to set the background, I want to ask the question, what are the major species of stink bugs in North Carolina corn? We could talk for a while about this since there's many, but really brown stink bug makes up 97 to 98% of the species. We do have rare cases where we see green stink bugs sometimes, or southern green stink bugs that predominate, but these are much more the exception rather than the rule. So today when we're talking about stink bugs in corn, just keep in mind we're primarily talking about one stink bug species. Now there's really, when we're talking about brown stink bugs, we're talking about Euschistus the genus. There's lots of different Euschistus species, but the main one that predominates is Euschistus cervus. So if you see a brown stink bug, it's probably in that genus, it's probably a Euschistus cervus, and there's really only one other thing that you could confuse it with, and that's a spine soldier bug. And these are actually predatory stink bugs that prey on a lot of pests in our cornfield, so it would not be a good idea to go out and sample stink bugs and confuse this with the pest brown stink bug. There's a pretty easy way you can tell if it's a spine soldier bug, as you can tell from the name spine soldier bug. You can look for the presence of the spine on the back of the insect, and you also see how the wings protrude out of the back of that really hard shell on the back of the stink bug. That's sort of a dead giveaway. But if you still can't tell if it's a brown stink bug or a spine soldier bug, you can look at their beak, and you notice the single thin beak of the brown stink bug that's built for piercing plant material, and you look at the double thick beak of the spine soldier bug that's made for piercing the hard shell of other insects and sucking out their content. Now we can find stink bugs on corn throughout the season, but there's really only 3 major times when we need to be sampling and watching and possibly treating for stink bugs in corn. And that's because these are the times in the corn plant's life when it's most susceptible to damage from stink bugs. And that's during the early vegetative stages, where damage to the corn can cause stunting, it can cause tillering, cause yield reductions later on in the season, perhaps a delay in development of the corn. The pre-tasseling stage, that's when the ear is forming before it's even pushed out, and damage during this stage can lead to that banana or boomerang ear feature. And then during the reproductive stages, when it can destroy kernels or perhaps transmit mycotoxins, you can see some of the starburst aflatoxin developing on the ear there. We have thresholds for each of these stages, and we're actually not changing any of these thresholds, but what we've done is Arun has taken a lot of data on sampling of the corn plant, and we've taken these thresholds and we've modified them to fit his sampling plan. So all the modifications you're going to see for thresholds today actually come from published studies in the literature on these thresholds. And to apply thresholds, we need to know what insect we're dealing with. We've talked about identification, but we need to know how many there are. And you need to know where to find these stink bugs. So where do we look for them in corn? We know places where stink bugs like to be, and one of those is in no-till fields with heavy cover. These can possibly provide overwintering sites for the stink bugs. They can provide cover from other things that might want to eat them, but they're especially problematic because when we plant corn into fields with heavy cover, sometimes those press wheels don't close the furrow up, especially if we're on some stiffer land. The problem with that is that stink bugs might be able to reach the growing point a little bit earlier. These are some data that Arun took on the growing point of corn, and he's got a little line there that shows where the soil line is. And notice that at V2, the growing point of that plant is actually below the soil surface. It doesn't even reach the soil surface until V5. So even though the plant's above the ground, the growing point is still below the ground. We really want to avoid that growing point from getting hit, because when it hits that growing point, that's what causes the tillering. If you have a field where the furrows are open, the stink bug can reach that growing point much more easily. We also know that we can find stink bugs at the wheat-corn interface. That's because brown stink bugs develop a generation in wheat often and then can move into adjacent corn. This is really more of an eastern North Carolina problem, but really anywhere wheat is grown, this could be an issue. And I should mention that all of our sampling recommendations that we're mentioning today do not apply to the wheat-corn problem. That wheat-corn problem is special. They move from wheat to corn right after the wheat's being harvested. We have some data showing this. This is a sampling study done on a wheat field next to a corn field. Anytime you see a dark dot, that's where the stink bugs were. And you notice those dark dots are all on the left side of the picture. Those stink bugs are still in wheat just prior to the corn harvest. Well, once the wheat's harvested, you notice that they start to move into the corn on the edges first, and then they move throughout the field. And these stink bugs can be found anywhere on the corn plant, especially as they're moving. So all these sampling recommendations we're going to talk about do not apply to this special case of stink bugs moving from wheat to corn. These are some data that Arun generated on overwintering stink bugs. And these are stink bugs that he found on the ground in a 25 square meter sample. And notice how many more he found behind last year's soybeans compared to last year's corn in 2016. We didn't exactly see the same trends in 2017, but you notice that even though there was much more variation, the most stink bugs were found. You see those top two dots over there. The most stink bugs were found behind last year's soybeans. This also illustrates that this is no guarantee. No 2 years are the same. Just because you're near good overwintering sites, that's not necessarily a guarantee that stink bugs are going to move into corn. And just because you're far away from overwintering sites, that doesn't necessarily mean that you're safe from stink bugs. But these are sort of just general trends that we can pick out. And even if we take soybeans out of the picture, if you plant corn near good overwintering sites such as woods, stink bugs can be an issue. If we think about when corn is planted, it's planted early in the season. These stink bugs are moving from their overwintering sites looking for things to feed on early season, and that could potentially be seedling corn. Where should you look for stink bugs in the field? We've known for a while that stink bugs are edge-associated species. These are some data from Georgia that demonstrate that. You can see these lighter colored areas towards the edge of the field, and that's where he found most of the stink bugs in this study. You notice that they're very patchy. Stink bugs are known for this, unlike some other pests that are distributed almost evenly throughout the field. A good example of that would be corn earworm. You could walk in the middle of a cornfield and probably find about as many earworms as you could as the edge. Stink bugs are very much edge-associated species. So if you want to see if stink bugs are there, a good place to check would be starting at the edge first and then moving into the middle of the field. And finally, stink bugs not only prefer the edges of the fields, but they also prefer different parts of the corn plant. And Arun has given us some photos of where we can find stink bugs gathered in corn. Sometimes they'll feed on the base of the plants. Here's some stink bugs feeding on brace roots. Here's a stink bug hanging out on a leaf. Here's some stink bugs on the stalk, likely the ears developing in this part here. And here's a stink bug feeding on the kernels of the corn plant itself. And Arun has actually generated some data on this. So he's got 2 years of data, and he's broken out where these stink bugs are on the different growth stages. And we can see that early on in the corn growth stage during V4, most of those stink bugs are found on the stem, and they don't move to the whorl of the plant until later on, V8, V12. And then when we look at the reproductive corn stages, we see that stink bugs are found on the stem in the early reproductive stages and then move to the ear or ear zone as the corn develops throughout these different reproductive growth stages. We can also look at this a different way. We can look at where stink bugs are vertically as well as horizontally on the plant. So horizontally, you can see in the different x-axes here, and this is just 2016 data to illustrate it. And then vertically, you can see on the y-axis there. And each one of those little dots is where Arun found a stink bug. And you notice that they have a tendency to congregate or disperse during different growth stages. More congregation towards the base of the plant in V4, a little bit more dispersal through V8 and V12. And then as the corn is approaching tasseling at V14, they're starting to tighten up a little bit more around where that ear is developing. And indeed, as we saw before in R1, they're starting to congregate around that ear zone, and then R2 through R4, they're really congregated on the ear. So Arun's taken this information and he's looked at the relative frequency of where these stink bugs are found with the hope of being able to change our scouting recommendation from scouting across the entire plant to where these stink bugs are congregated at these different growth stages. And so these are data that he has taken from 2016 and 2017, and he's got this distribution in relation to the ear leaf, and he's noted the frequency of where these stink bugs are either above or below that ear leaf. And what Arun did was he used a number of different criteria, looking at different measurements of variation, different statistical measurements, and then relative net precision, which takes into account efficiency of the sampler while they're searching the plant, as well as the precision of finding the insect. And he settled on this right here, ZP1M1M2. That's basically saying that the most cost-effective scouting method for sampling stink bugs at R1, R2, and R4 was right on the ear, and the stem 1 leaf above, and the stem 2 leaves below the ear, which is outlined in the orange on the plant that you see here. Now we've also taken this information and we've converted it into a sequential sampling plan. And I'm just showing you this graph here to show you what a sequential sampling plan is. So for a hypothetical insect in a normal sampling plan, what we might tell you to do would be to walk out in 20 places in the field. If you find this many insect across those 20 places, you need to treat. With a sequential sampling plan, what you can do is you might be able to stop sampling before you visit all 20 sites. So with a sequential sampling plan, if you find a number of insects or below that, you can stop sampling. If you find more insects than that, you can treat before you need to sample all those places. It's only with those borderline cases that you need to continue sampling. And so that's what we've done here with these stink bugs on corn. We've come up with a sampling plan. The number of samples that you're going to need to take in each field is going to depend on the field size and the characteristics of that given field. So for this hypothetical example, we've said, in this particular cornfield, begin at 10 places at the field and take a 10 plant sample. So for at each spot, you're going to take a 10 plant sample, and then you're going to do that 10 times. We've also broken this out into 3 different growth stages: V1 to V6, V14 to tasseling, and R1 to R4. The reason we don't recommend sampling V7 to V13 is because we don't think that the stink bug can injure corn at these stages. So I would say that you don't even need to worry about sampling. I also want you to keep in mind that this is for the situations where corn is not next to wheat. When corn is next to wheat, the stink bugs seem to be found very much more in the whorl and distributed at different places in the plant. But for most of our situations that are not near wheat, we can only sample particular areas of the plant. So for V1 to V6, we would say that you only need to sample the stalk below the lowest green leaf. And we specify the lowest green leaf because by the time the plants are V6, some of those lower leaves could have died away. So all you need to do is look below that lower leaf. For the sequential sampling plan, if you find 6 stink bugs in 100 plants or fewer, you can stop sampling. If you find 13 or more, you need to treat. And then between there, you probably need to take some more samples to see whether or not you've hit threshold or not. For V14 to tasseling, you only need to sample 1 leaf above and 1 leaf below the primary ear. Now, how are you going to find that primary ear if it's not pushed out yet? The corn plant's not even tasseled yet. You probably need to cut open the stalk to look, because although the ear oftentimes forms around the 14th leaf, it depends on the hybrid and different environmental growth conditions. In addition, as the plant approaches tasseling, it's growing very rapidly, putting on nearly a leaf a day. And so the location of that from the top is going to vary depending on what day you're sampling. But you can figure out where that ear is forming based on splitting the stalk. With our partial plant sampling, if you sample in that area and you find 12 stink bugs in 100 plants, you can quit sampling. You don't need to treat. If you find 18 or more, you do need to treat. And in that middle area, you probably need to continue sampling. And similarly, from R1 to R4, we have an area of the plant that you can sample 1 leaf above and 2 leaves below the primary ear. So we've added a leaf here, and also the stem area below those leaves. If you find 35 in 100 plants, you can stop sampling. If you find 52 or more, you need to treat. And then in that middle gray area, you need to continue sampling. Our current insecticide recommendations have not changed. We recommend bifenthrin for brown stink bugs. This is the most effective insecticide in many crops, and that includes corn. We've taken stink bugs out of corn, put them in a vial bioassay against other pyrethroids. Bifenthrin is more potent than cyfluthrin, the active ingredient in Baythroid and Tombstone. It's also more potent than lambda-cyhalothrin, the active ingredient in Karate or Warrior. So bifenthrin is really the pyrethroid of choice. And we'll put all these different recommendations up on the NC State portal so that you can go view them for your reference, because although you'll be able to take fewer samples in the field and save time in the field, there are more difficult numbers to remember than before. I want to end this presentation a little bit by giving a little bit of an update of corn earworm and Bt resistance. I want to preface everything I'm going to say with a fact, and that fact is that corn earworm is rarely a yield-limiting pest in timely planted corn in the southeastern U.S. We've done these studies in both North and South Carolina. They've replicated these in the Mid-South and have found much the same thing. If we plant our corn on time and have good growing conditions, corn earworm will eat those kernels in the tip, but the plant can compensate perhaps by filling out kernels near the base a little bit fuller. It can compensate for that injury, and we won't see a yield reduction from the earworm. So corn earworm rarely limits yield. Well, you say, I've had non-Bt hybrids that yielded worse than Bt hybrids. Well, that may be true, but what I would ask you is it 100% genetically pure? Or is it, as the breeders tell us, 95% genetically similar? Like I like to say, I'm 98% genetically similar to a baboon, but I like to think that I'm very different than a baboon. And I think much is the same with corn genetics as well. Just because a hybrid is very closely related doesn't make an exact match. I would also say that corn earworm can be a yield-limiting pest, certainly in situations where there are other stressors in the environment like drought or under heavy pest pressure. These things can cause yield loss. And in addition, we do know that they can transmit mycotoxins, things like aflatoxins and fumonazins. The most recent studies have shown that these associations are very loose. Yes, insects such as earworms can transmit aflatoxins, but there are other greater overriding factors, probably things like genetics and environment, that play more strongly into aflatoxin. So I think from a yield perspective, in most of our corn earworm is just not a yield limiting problem. That being said, we are very concerned about resistance in Bt corn of this insect for a couple reasons. Number one is you're paying for these traits that are supposed to work against earworms. In fact, we're advertised to do so. So in my opinion, if you're paying for something, it should be working. And the second reason is that we're concerned about these insects moving from corn into cotton. A longtime entomologist, Dr. Stinner, in the 60s, wrote that he thinks that earworm populations in North Carolina would be reduced by 97% if corn were removed from the system. These things are essentially a factory that are generating insects that later move on into cotton and soybeans. And let's keep in mind that many of these Bt traits that we have in corn are related to Bt traits that are in cotton. So corn is probably acting as a filter. It's filtering out insects in the system, causing resistance that then causes us problems later on in cotton. And we do know that we have bollworm, which is the same insect. We do know that we have bollworm that's now resistant to Bt cotton. And it's likely a function of all the Bt corn that we have in the system. So with that background, let's shift back to corn. This student in my program, Shawnee Gundry, has looked at a number of different things across the state. And this is a cooperative project with many different county agents across the state where they planted paired Bt and non-Bt hybrids. And one of the things that Shawnee was able to do was she was able to go out in the fields and measure the amount of feeding in these different Bt hybrids relative to the non-Bt hybrids. Well, so what I've done is I've taken her data and I've compared the amount of feeding in the Bt, which is the VT Double Pro, to the non-Bt. So if there's a lot of feeding in the VT Double Pro compared to the non-Bt, we would expect the bar to be high. If there's relatively little feeding in the Double Pro compared to the non-Bt, we expect the bar to be low. So the higher bars, we think that there's probably more resistance there. It's breaking through the trait. If there's lower bars, we expect there's less resistance. So I'm going to focus in on the left side of the graph here. This is areas where the trait seems to be working. Notice that it seems to be working in many of our eastern locations: Bladen, Pamlico, Sampson, Edgecombe, Harnett, Onslow, but then we have Union County out there, so very geographically different. Well, let's go over to the right side of the bar, and I want to point out that anything over 1 means that there was actually more feeding in the Bt Double Pro relative to the non-Bt. Washington County, Gates County, Hertford County, Brunswick, Johnson, Terrell, Pasquotank, Pitt. So in many of these eastern counties, it looks like the Double Pro trait is failing as well. And if we just pick on one county here, Washington County, look at it, these 3 locations within the county, the range in how this trait's working. Anywhere from 50% effective all the way to totally not effective, more feeding in the Double Pro. relative to the non-BT. We've taken this feeding information and we've also looked at the proportion of infested ears. So we wanted to see, are ears more infested in the Double Pro compared to the non-BT, and how is that related to feeding in the Double Pro and the non-BT? And we've correlated and we found there's a very high correlation. This line basically says that as there's more feeding in the non-BT relative to the non-BT, there tend to be more ears that are infested relative in the BT relative to the non-BT. So these things are related. And we can also look at pupil exit holes and see much the same thing. As there's more feeding in the VT Double Pro relative to the non-BT, there tend to be more pupil exit holes as well. So that tells us if we see a lot of feeding in corn, we'll probably have a lot of insects that are exiting out of the ear, pupating in the ground, surviving on to infest crops later on in the season. Another thing Shawnee was interested in doing was bioassaying some of these populations on Cry1AC. Why do we choose the BT protein Cry1AC? We chose Cry1AC because this is the protein that's present in all the BT cotton that we grow. Things like Bollgard 2, things like Twin Link, things like Wide Strike, and even the newer varieties, the Bollgard 3 and the Wide Strike 3 varieties, they all have Cry1AC in them. And they're related to the Cry1A105 that's present in the VT Double Pro that we were looking at. We actually expected to see a correlation in survival with Cry1AC with feeding in Double Pro. That is to say, as feeding increased in VT Double Pro relative to non-BT, we also expected to see survival on this bioassay of Cry1AC to be correlated, but there was no correlation. So let's just look at these data. What she did was she screened them on a 95% lethal diagnostic dose. She determined that dose by using 4 susceptible colonies, putting them on Cry1AC and basically seeing how many of them lived or died. And then she took these populations from non-BT corn in different counties and she ran them on this bioassay. So any population that we find below that line we consider susceptible. And there were 2 locations that were susceptible, one in Granville County, one in Washington County, but there were many locations where survival was more than expected, all the way up to over 70% of them surviving on a dose that should have been 95% lethal, and that occurred in Wake County. Much like we see with the feeding data, there's a range of susceptibility in these populations. So if we look at a single county, Washington County again, and we look at the locations 1, 2, and 3, we can see a range of survival all the way from very susceptible to not susceptible at all. So what that tells me is the susceptibility profiles of these things are all over the board. You don't know if the earworms that show up in your cornfield are going to be resistant to this BT or not. You don't know if the earworms that are produced from the corn are going to survive well on your BT cotton or not. We're sort of shooting in the dark. And I think based on this graph and the feeding data we've seen, it's probably best to assume that there's at least some level of resistance that's out there. Let's also keep in mind that this resistance is not either all or none. There's a range of survival, there's a range of how these things behave, and a lot is dependent on the environment. You know, is it going to be a good earworm year? Are there going to be a lot of earworms out there? Are we going to have environmental conditions that favor their survival? So we're not really sure what we're going to see with these things in corn next year, but I guarantee you there will be some survival on BT corn out there next year. If you have any questions, you can email me. Another good way to get a hold of me is on Twitter. There's my Twitter handle right there. Thank you for your time and patience. Again, I'm sorry I couldn't be there today. Thank you.